So I'm going to talk to you about NMDC's standardized metagenomics workflows and the pipeline that we've created. My name is Karen Davenport, and my background is with genomics, metagenomics, and bioinformatics. And I'll share with you everything I can. So why would you want to use a standardized workflow? Improvements to bioinformatics are important for the advancement of science, but the number of tools that are out there developed for metagenomics is exponential. Just the number of tools out there to map sequences, one sequence to another or to a database, is an exponential graph. It's not uncommon for a graduate student to create a tool that's developed just for their particular project, and it's never used by anybody else. So the data sets that are out there and the data products that are out there aren't comparable. We're not able to compare across studies. And so the idea of using bioinformatics workflows that are standardized allows you to utilize data from other studies as well. It also helps you keep track of what you did with your own samples and make sure that they were all processed in exactly the same way. So the NMDC standardized workflows incorporate state-of-the-art bioinformatics that were developed for all types of multi-omics data. We're going to focus for this particular talk just on the metagenomics workflows, and those include reads QC, read-based taxonomy classification, assembly, annotation, and metagenome assembled genomes. All of these workflows were developed for high-quality short-read data from the Illumina platform. So there are 5 different workflows, as you can see here in blue, and these what we've done is generate a pipeline, create a pipeline that takes raw Illumina data, and it's linking the output from one process to the input of another process so that the user can simply put in raw sequencing data, and it will take it through an end-to-end pipeline through all of these different analyses. So while all of the workflows are available for download through GitHub as Docker containers, we've also put all of them into this user-friendly interface as a publicly available resource, and it's called NMDC Edge. This is for researchers who are not comfortable with command line tools, or for those who don't have the computational resources at their institution to run the Docker containers. So we're going to go through each of these individually. Reads QC, it takes FASTQ format as the data input. If you're not familiar with FASTQ format, the data comes off of the sequencer with not only the sequence itself, but also with a quality score for every nucleotide in that particular read. The tools here in this pipeline come from BB Tools, and the output is also FASTQ. And so this is a set of clean reads, and you also get some QC statistics. So what does QC do? What does data QC include? It can be trimming, cutting off part of a read because it's low quality, or because these are adapters or primers as part of the sequencing process. Or it can be that that the bases weren't called as A, T, G, or C, they were called as ambiguity codes. So those are all things that you could trim a read by. You can also filter reads by, is this read just adapter or just bi-X or primers? These are things that are typically part of the sequencing process. If you trim a read and it becomes very short because most of the read was low quality or low complexity, it might be, only 25 or 30 bases long, And so you might want to discard all of the reads that are less than 50 bases. You can filter a whole read if the ambiguity code is a certain percent level, meaning that most of the read or some percentage of the read is not called as A, T, G, or C. You can, the same is true as if you have a long stretch of mononucleotide or dinucleotide sequence, so that the complexity of that sequence is very low. Those are typically not realistic sequences and should be removed before downstream analysis. You can also remove host, whether that's human or mouse or other common examples. So for example, if you're looking at a soil sample and you know that cattle have been in that field, you might want to remove anything that matches the cow genome. And then of course you can remove by quality score of the entire read. Read-based taxonomy classification takes in Illumina data. We highly recommend that you use clean reads from the previous workflow, because why would you want to try to classify reads that are low quality or adapters? That just wastes computational time and effort. There are probably dozens of classification tools for short read sequencing data, and there's no one perfect tool. Our workflow takes in the FASTQ files and utilizes 3 different classification tools or profilers, and the output includes both tabular text files and interactive chronoplots for each of the tools at 3 different levels of taxonomy. If you're not familiar with chronoplots, the Chrono is an open source software package for interactive visualization of genomic sequences for metagenomic samples. So let's talk about taxonomy classification of metagenomes for a few minutes. Most taxonomy classifiers generally work by taking a read and mapping it against a reference database. And whatever the best match is in the database is what is called the taxonomic classification for that particular read. Most of them have databases that are developed for the particular algorithm that that tool uses. So there are lots of problems with that. There are limitations. It's highly dependent on the database, and our databases will always be incomplete. This will always be a problem. Only a tiny portion of of the organisms on the planet have been sequenced. And so if you try to map a read to something that isn't in the database, it isn't classified, or maybe it sort of matches something in the database, but isn't a really good match, then it's up to whether or not that algorithm calls that particular classification. There are also incorrect entries in the database, which is one of the things that Bill mentioned earlier. And then of course, there are issues with taxonomy itself. There are name confusions, there are name changes for organisms, and there are synonyms. And all of these create problems with classifying reads. So as I mentioned just a moment ago, each of the different tools uses a different database and algorithm. And so these tools produce different results. In our opinion, it's best to use a combination of tools because there is not one best tool. And we use a combination of tools in our workflow, 3 different tools. Kraken is very sensitive, Centrifuge is a mid-level tool, and Gotcha is very specific. A sensitive tool or test allows you to rule out the presence of an organism if the result is negative. So you get very few false negatives, but you can get a lot of false positives. The flip side of that coin is a very specific tool allows you to rule in the presence of an organism if the result is positive. So you get very few false positives, but you can get false negatives. And so that's why it's best to use a combination of tools. Other considerations for taxonomic classifiers are, when do you call an organism present? If you have just one read from your sequencing run, or do you have to have many reads? Well, what if those reads are all stacked up in one location in the genome? Or do you only call it present if you have broad coverage across a genome? And then, of course, there's contamination and eukaryotic or host cross similarities. And how do you determine abundance? The same sort of problem. Can you do it with numbers of reads that are classified? Well, remember, different taxonomic tools classify things in different ways, and so that's a difficult call. And of course, then there are all the database issues. Some species have been sampled and sequenced many more times than others, and are highly prevalent in the databases, and some are not there at all. There are errors in sequence from the sequencer itself. There are errors in taxonomic classification in the database, and just inconsistencies. So this is not a straightforward one-stop shop. You can use one tool and get all of the answers for everything that's in your metagenome. Assembly. Assembly takes in preferably clean Illumina data in FASTQ format. The tools that we use here are Metaspades and BB Tools, and the output is in a file of assembled contigs in FASTA format, which means that you have the nucleotide sequence of the contigs, but there aren't quality scores associated with each of the bases. And then you get some assembly statistics as well. So putting together a genome is like putting together a puzzle. Sequencing is the process of taking a genome and breaking it into millions of pieces and to be sequenced, and assembly is just trying to put all those pieces back together. Metagenome assembly is like putting together many puzzles at the same time. And it's much more complex. And you can have genomes that are similar, which make it even more complicated. So let's talk for a minute about contigs versus scaffolds. I know Bill brought this up. Most DNA is sheared to somewhere between 300 and 600 base pairs for sequencing for short read data. It's sequenced so that it sequences off both ends. That's the Illumina sequencing process. And so you have the data comes out in two files, a forward and reverse. So all the orange pieces here would be in the forward file, and all the blue pieces would be in the reverse file. And they're numbered so that they match, so that you know this is a pair. When you put all these pieces together, you can get a contig that is a consensus sequence from a group of reads. A scaffold, however, is when you have two or more contigs put together, and you know the order and the orientation of those contigs, but the middle sequence, there's a gap where you don't know what that sequence is. You might even know how many bases go there, but you don't know the exact sequence. And an example of what might create a scaffold would be where you've got a read where one end of the fragment is in this contig, and the other end of the contig, at the other end of the fragment is in this contig, and this middle section is unknown. So what you get in the end, your assembly, is an approximation of the original sequenced material based on the data that you generated. There are different types of assemblers. One is called a greedy extension, where it starts with a single read, and it then maps another read, all reads to it individually, to try to extend the known sequence to create contigs. and then it repeats with another one and another one. An overlap layout maps all reads to all other reads to find pairwise overlaps to then create a layout graph to create contigs. And lastly, there's the De Bruin graph sequencers that takes all of the reads and breaks the sequences in those reads into smaller pieces of all the same size called k-mers. And in this case, this example, the k-mers are 4 nucleotides long. A rough estimate is to use a k-mer that's maybe 3/4 the length of your read, but that varies and is generated automatically by many of the assemblers. Anyway, then it creates k-mer graphs, and then it tracks paths through those k-mer graphs to create the contigs. This particular way is computationally much less intensive than either of these methods, and therefore it's the only realistic type of assembler to use with the mountains of Illumina data that are generated for genomes and metagenomes today. So why is it difficult to assemble a genome? Well, you've got gaps, misassemblies, and incorrect sequences due to lots of issues. There may be sample prep issues, issues with the integrity of your DNA and RNA, or RNA, depending on what you're working with, or contamination. There are issues with the sequencing technologies themselves. Although Illumina is a very mature technology, the longer Illumina read lengths tend to have lower quality at the end, and so that's an issue. There are different error rates and error types. How many errors per how many base pairs? And are those errors insertions or deletions, or are they homopolymer repeats? Again, the quality level for the different sequencing technologies, and the level of coverage that you can get, or the number of reads per sequence material is important when you're choosing an assembler. And of course, there are sequencing biases. Illumina used to not go so well, not sequence so well through high GC organisms. Again, there are different assembler types that we talked about just before, but you need to choose an assembler for the particular sequencing technology and the amount of data that you have. And then, of course, there are structural issues with the genome itself. There are long repeats that Illumina data will never get through. There are tandem repeats, which means several repeats of the same thing, anywhere between 2 and 20 repeats of the same thing, and short read assemblers can't get through those tandem repeats. GC content, again, high or low, and structural variance within the genomes can create assembly problems. If you have the opportunity to add another technology to your short read data, you can get complete or near complete genomes. Some of these technologies include an optical map, long read assemblers, meaning PacBio or nanopore data, or a high-C data, which links together sequences that are next to each other or associated with each other in the nucleus of the cell itself before you do the sequencing. All of these can help, but NMDC does not have these capabilities to do hybrid type assemblies yet. Could be coming. But the Metaspades is the assembler that NMDC uses for its assembly workflow, and it's a very effective metagenome assembler. But I want you to look at something here. This is from a study looking at different metagenome assemblers, these Metaspades, MegaHit, IDBA, and RayMeta, with 4 different levels of complexity for metagenomes. This is a small synthetic metagenome. This is from the Human Microbiome Project, which is more complex. This is a marine sample, which is more complex still. And here's a soil sample, which is the most complex. If you look at the fraction of reads aligned back to the assembly, we can use this percentage as an estimation, a kind of a rule of thumb about how well the data assembled. You can see with this less complex metagenome here, the least complex, that all of the assemblers hit, mid to high 90s for a fraction of, or at least 90s, for the fraction of reads that map back to the assembled contigs. If you look at the next one, as you go down, then this begins to drop, the amount of reads that map back to the assembled contigs. And as you go further down, you see that the more complex the metagenome, the less capable we are of actually assembling all of the data. But Metaspades is still the most effective metagenome assembler we have at present. Bill just gave you a whole talk on metagenome annotation. What our pipeline takes in is the assembled contigs in FASTA format, and the recommended input, of course, is from the Metaspades assembly. Our workflow has a whole suite of tools for identifying gene or structure or function. And the output are a structural annotation file, a functional annotation file, several files from the separate tools and databases. And these are in tabular form as either TSV or in GFF format. So these are all of the different tools that, I believe these are all the different tools that are used in the NMDC annotation pipeline. And the final structural and functional annotation files are kept separate. But we're combining all of the output from these to create these final files. Metagenome assembled genomes. The input, of course, is the assembled contigs, and a read mapping file from the assembly. Remember, that's where we get the percent reads mapped back. It's a way to calculate the level of coverage per contig, and that helps them sort these out. The tools, there are several tools that are used to bin the contigs and then refine those bins to give us the mags. And the output are the bins that are deemed high quality and medium quality, along with the summary statistics. So why is it difficult to get complete genomes from metagenomes? Well, you have lots of different organisms at lots of different levels of abundance. They may share regions. We may have several strains of the same type of organism from the same genus. These will have different levels of coverages, and so we just get different levels of completeness. But what we do get are draft genomes, and we can get what to me seems like an amazing number at this point. The binning process uses MetaBat2, which is more computationally intense than MetaBat1, but is more effective. It's sorting, so you have all of the contigs that you've sequenced here, and here are all of your reads, and here are your contigs. It's sorting those contigs by tetranucleotide frequency, as well as abundance, and then a calculated pairwise matrix to then give you bins with contigs that should be from the same genome. Each resulting bin is assessed for completeness. I believe it has to be above 90 or 95% to be called a high quality bin, with less than 5% contamination. And for a medium quality bin, it has to be considered 50% complete with less than 10% contamination. So why would you want to use the NMDC workflows? Well, these are tried and true bioinformatics tools, and they've been carefully researched. They've been tested at many, many different institutions from different sequencers and sample types. We have these available to you through shared computing resources so that you don't have to download the tools or all the databases that go with these tools. And you don't have to have access to your own computing clusters. It also allows for direct comparisons with other omics data sets. And because all of the data in the NMDC data portal has been put through these NMDC omics workflows. And this is where you can find other, this is the NMDC data portal, which I think you learned about yesterday, and you can find other data sets that might be related to yours that might be an interesting comparison to your own. The data for this afternoon session are from the Spruce study. This study and its data products are in the NMDC data portal. Here are some links that might be of interest to you that relate to the NMDC workflows. Of course, this is where we're going to go this afternoon. And if you're interested in becoming a beta tester, this is still a beta product. You can email this address right here. We are looking for beta testers. And with that, I will stop and take questions.
Thank you, Karen. There are a couple of questions that have popped up. The first one has to do with input into the process, and I think I'm getting this question correct. So the question that was asked was, can a FASTA file be used for input instead of a FASTQ, I believe for the read files?
Not at the moment. There are ways around that, but that's not what's in the pipelines, the workflows at present. It has to be, well, it depends. It depends on the workflow. If you wanted to input FASTA files just for annotation, you could do that.
Okay, but for if you wanted to actually do the assembly part of the pipeline, you would need to have FASTQ.
You have to have FASTQ files.
Okay, another question was, the metagenome workflow does not include read-based annotation, i.e. human N. Why is that? Are there major issues with read-based annotation that assembly solves?
You know, assembly gives you much bigger pieces, much longer pieces of the puzzle, you know, of the puzzle are put together so that you can find more genes. We're talking about short read data where, you know, most of the reads are, you know, 200, maybe 300 base pairs long. And those are just much more difficult to annotate.
Okay, here's a question about the parameters. It says, how much do the customizable parameters affect how comparable the analysis is, even though the tools used are the same?
Well, you're not allowed to play with the parameters in these workflows. That's part of the standardization. You know, we've done our best to optimize all of these tools, and that's what allows you to have comparable results. That, you know, you're running the same tools with the same parameters, and then you can compare the data products.
Okay, next question is, when we use the NMDC tools, do we need to worry about memory usage? Some tools may need more memory than others and may cause memory explosion.
We've had some memory explosions as we go through this process. And yes, that is absolutely true. Annotation is the most computationally intensive and memory intensive. And we've had things, you know, your run might fail depending on how much data you have there. We're constantly working to improve how these pipelines and how these workflows, individual workflows work. And we are asking for more resources for this publicly available version of this so that we can increase our capabilities to offer to you.
Okay, I don't see any other questions that I might have missed in the Discord. And I don't think there are any more in the chat. So we can hand it back over to you, Montana or Darian.